Following the steps of replication of pathogenic mycobacteria within the macrophages, the infected cell migrates into tissues where a granuloma is formed with additional immune cells. Majority of the bacteria and surrounding host tissue is destroyed by the granuloma. The bacteria which survived exists in a latent state, meaning it is dormant in the macrophages. The latent phase is a state that occurs after an individual has been exposed to the tuberculosis bacterium, the infection has been established, and an immune response has been generated to control the pathogen and force it into an inactive stage. In contrast to patients with active TB, individuals with latent TB do not transmit the disease and do not pose a public health risk. Several mechanisms are used by the MTB in order to withstand the harsh conditions that they are exposed to. These mechanisms can be divided up into three pathways that ultimately lead to the survival of the bacteria: metabolic changes, cell wall changes, and signal transduction. Involved in metabolic changes, ICL is an enzyme primarily responsible for redirecting acetyl-CoA from beta oxidation into the glyoxylate shunt. This enables Mycobacterium to obtain carbon in harsh conditions inside macrophages by metabolizing solely acetate and fatty acids. The MTB phosphorylates and thus decreases the activity of PCAA, or cyclopropane synthase enzyme, which is essential for the formation of the phagosome maturation block and the production of mycolic acid, an essential and important component of the cell wall. In turn, this contributes to the survival of MTB in the host macrophages during the latent stage. Signal transduction, which is used by MTB to overcome hypoxia, is regulated by two component systems composed of two sensors, DOS-T and DEV-S, and their response regulator, DEV-R. As we can see in the video, the first step is the autophosphorylation of DOS-T, which detects early oxygen depletion and binds to ATP with its ATP binding site. A kinase region is then responsible for phosphorylating the 54th residue for response regulator, DEV-R, aspartic acid. When hypoxia becomes more severe, the same process explained previously occurs with a second sensor, DEV-S. Due to the lack of O2 bound to the protein kinase regions of DEV-S, these regions get autophosphorylated. Consequently, DEV-S takes over and continues the phosphorylation of DEV-R. DEV-R is the second part of this system. When phosphorylated by DEV-S at its 54th residue, it becomes activated in the promoter region and begins to react in response to hypoxia. DEV-R is responsible for regulating a 48-member regulon, essentially meaning that upon activation, it initiates the transcription of a multitude of proteins that are necessary in TB's ability to become and stay dormant, as well as survive in these conditions. This is achieved by the binding of DEV-R to an RNA polymerase. A complex is formed and cooperatively binds to DNA, initiating transcription of the proteins under DEV-R's control.